Hello students, today I am going to take a topic cell cycle and in that particularly interphase and mitosis. And I am Geeta, I am botany faculty. I will be taking up that topic for you. In this cell cycle topic, you will be getting at least 4 questions, 3 to 4 questions in the coming NEET. So you have to prepare thoroughly for that. Let us see the cell cycle first. In the cell cycle, you know that the cell cycle is having mainly two phases. One is interphase and another one is M phase. So in the interphase, we have G1, G2 and S phase. These three will be there in the interphase. So interphase will be comprising the first G1 phase, then S phase, then G2 phase. And these three together, they will be called as interphase in the cell cycle. And then M phase, it is the divisional phase. Divisional phase, and it is also called as M phase, mitotic phase. So in the cell cycle of a normal eukaryotic cell, any cell you can take, human cell if you take, generally if you take the human cell, 24 hours will be the time period which is required to complete one cell cycle. So in that, the interphase will be taking almost 95% of the time, and only just 5% of the time will be given, even less than 5% of the time will be given for the M phase or divisional phase. So almost 95% of the cell cycle will be utilized by the interphase only in the human cells, or in other cells also it will take the same time. But M phase will be taking just 1 hour. In case of human cell, it will take only 1 hour, and remaining 23 hours is there for the interphase. So this is how the cell cycle will be divided into two main phases. One is the interphase and another one is the divisional phase or M phase. In the interphase, we have interphase is subdivided into G1, S and G2 phases. In this G1 phase, RNA and protein synthesis will happen. And the cell will be growing. Cell growth will be happening, and it is also called as gap 1 phase or first growth phase. And S phase is called synthesis phase because here DNA synthesis or replication occurs. DNA synthesis or replication of DNA occurs in S phase. Along with that, centriole duplication also occurs. Centrioles duplication occurs in the S phase. This is the most important phase of the interphase, and it is the important one in the interphase alone, and it takes long time. Then the G2 phase is called as second gap phase, and here the RNA and protein synthesis will be continued here also. RNA and protein synthesis will be continued, and this causes even the growth of the cell. Cell growth will be continued even in the G2. But out of all these, S phase will take long time among all the three interphase phases. S phase is the important phase because the DNA, one parental DNA will be replicating and it will be forming two daughter DNAs. And centrioles, in animal cells, centrioles duplication will happen in the S phase. So here G1 and G2, the cell growth will be continued in terms of RNA, protein synthesis and cell organelle synthesis, etc. But most of the cell organelles will be synthesized in the G1 phase only. Few cell organelles like mitochondria, they will be synthesized in the G2 phase. This is the interphase which is taking almost 23 hours in human cell. And it is the first phase. Actually, the cell will be taking rest. We used to think before, but it is the metabolically active phase, interphase, and the cell is preparing itself to undergo the division in this. So that is the reason why it will be synthesizing the RNA, proteins, ATP energy, cell organelles, multiplication, cytoplasmic contents increase, nuclear contents increase. All these things will be happening in the interphase. Now, the interphase is actually called as metabolically active phase, but the cell will not divide. And it is subdivided into G1, S and G2 phases. G1 and G2, they are concerned with the growth, and S phase is concerned with the synthesis of the DNA or replication of DNA, and in animal cells, centriole duplication occurs here. Next one, after the interphase in cell cycle, we have the mitosis, M phase, mitosis phase, or it is also called as divisional phase. In this mitosis, you can see that the interphase chromosomes will be like chromatin. In interphase, the genetic material will be in the form of uncondensed state and it is called as chromatin. Whereas in the mitosis we subdivide it into prophase, metaphase, anaphase, telophase. So here the mitosis is divided into 4 subphases: prophase, metaphase, anaphase and telophase. So chromatin will undergo the condensation in the prophase, and you can see that chromosomes are having the 2 chromatids very clearly in the prophase. And in the metaphase, the chromosomes are positioned in the center of the cell and this nuclear envelope disappears and this forms the spindle. Spindle apparatus is formed and chromosomes are aligned on the equator in the metaphase. In anaphase, this type of chromosomes, they split exactly in the middle with the centromere splitting, and it resulted in the formation of the daughter chromosomes, and that daughter chromosomes will move towards the opposite poles in anaphase. And then in telophase, here the moved daughter chromosomes to the respective poles, they would have formed the nuclear envelope around them, and all the cell organelles which got disappeared here, they'll be reappearing here, and it results in the formation of 2 daughter nuclei. These 4 are the phases of karyokinesis. Karyokinesis is nothing but nuclear division. Here 1 nucleus is there and it is dividing finally. By the end of the 4th phase, 2 daughter nuclei are formed. And after this, the cytokinesis occurs. Cytokinesis occurs and it divides the cytoplasm of the cell, single parent cell, into 2 daughter cells. So ultimately, after the mitosis, karyokinesis and cytokinesis, here you will be getting the 2 daughter cells. So mitosis results in the formation of 2 daughter cells only by the end of that. But it happens by 2 phases again, karyokinesis and cytokinesis. Karyokinesis is having 4 subphases: prophase, metaphase, anaphase, telophase, and cytokinesis occurs after that and it forms the 2 daughter cells. So clearly it says that mitosis is having 2 phases, particularly karyokinesis and cytokinesis. This karyokinesis is again subdivided into prophase, metaphase, anaphase and telophase. So clearly in this picture you can see that interphase will be having the chromatin and mitosis having the karyokinesis and cytokinesis. In the karyokinesis we have the subphases like prophase, metaphase, anaphase and telophase. After that, cytokinesis occurs once and it divides 1 parent cell into 2 daughter cells. Let us see the next one. The major events of these subphases. Prophase. You know that 4 phases are there in the karyokinesis and among them the first is the prophase. And this prophase is characterized by, the main characters of the prophase are: condensation of chromatin occurs. Condensation of chromatin into chromosomes. That is the first event in the prophase. The chromatin which was there, elongated form, that will undergo the dehydration and it will undergo the further coiling, condensation, reduced in the length, and they become the rod-shaped chromosomes. So that is the first step in the prophase, which is marked by the beginning of the condensation of the chromatin. Then after this, in animal cells, centrioles move towards opposite poles in cytoplasm. Already this centrioles got duplicated in S phase. So that centrioles which got duplicated in the S phase, they will move or migrate towards opposite poles in the cytoplasm. This event will happen in the prophase. The third event which will happen in the prophase will be ER, Golgi, endoplasmic reticulum and Golgi complex, nucleolus and nuclear envelope. Disappears. So the prophase end will be marked by the disappearance of ER, endoplasmic reticulum, Golgi complex, nucleolus and nuclear envelope. So these three are the major events which will happen in the prophase. Condensation of chromatin into rod-shaped chromosomes. Then in animal cells only, the centrioles which are duplicated in the S phase, they will move towards opposite poles in the cytoplasm. And in all the cells, eukaryotic cells, ER, endoplasmic reticulum, Golgi complex, nucleolus and nuclear envelope disappears by the end of the prophase. So that once the nuclear envelope disappears, the chromosomes which are formed already, they will be released or scattered into the cytoplasm because there is no membrane around the nucleus. So they will be released into the cytoplasm by the end of the prophase. Now the chromosomes are present in the cytoplasm. Next phase is the metaphase. Metaphase is characterized by mainly two events. On the chromosomes particularly, the two chromatids which are there, sister chromatids which are already there, they will not be seen very clearly in the prophase, but in metaphase we can see the structure of the chromosome very clearly. So morphology of the chromosome can be seen very clearly here. Morphology of the chromosomes can be studied here. Because the condensation would have been completed by the end of the prophase. Now, each chromosome will be having roughly this type of structure, the metaphase chromosome. And now, these two are called as sister chromatids because they belong to one chromosome only. This is called as centromere. On the centromere, now in metaphase what happens is, on the centromere, both the sides surface two disc-shaped structures will be attached in the metaphase and these are called as kinetochores. These kinetochores are meant for the attachment of spindle fibers. So the chromosomes, once they are released into the cytoplasm, okay, first on both the sides of the centromere, on either side of the centromere of all the chromosomes, two discs will be attached. They are called as kinetochores. And they serve as the connecting point or attachment sites for the spindle fibers. So spindle fibers which are formed already, they will be coming and attaching to the chromosomes from both the ends. So each chromosome is connected to both the poles by two different spindle fibers. From this pole, one spindle fiber, and from this pole, another spindle fiber will be connecting to the one chromosome. So that sister chromatids will be connected to two different poles. So this connection will be happening in the metaphase. First, kinetochores will come and attach, and to the kinetochore, spindle fibers will come and attach. The third event which happens here is chromosomes align at the center to form metaphase plate or equatorial plate. So the chromosomes already they are attached to the spindle fibers. With the help of the spindle fibers contraction and relaxation, all the chromosomes of that cell, they will be aligned exactly in the center or equator to form the metaphase plate. This kind of alignment is very important for the proper distribution of genetic material to the daughter cells. So these two are the major events. The condensation would have been completed in the beginning of the metaphase only, and we can study the clearly structure and morphology of the chromosome clearly in this. Then each chromosome will be connected to the spindle fibers from both the poles to the kinetochores. And with the help of the spindle fibers, all the chromosomes of a cell will be placed or aligned on the equator to form the metaphase plate or equatorial plate. These are the major events of the metaphase. Next phase is the anaphase. In the anaphase, centromere splits. Centromeres of all the chromosomes will be splitting and they form daughter chromosomes. And they form daughter chromosomes. Now, the daughter chromosome will be having only this type of structure. It will exactly split in the center, and the daughter chromosome will be having this type of structure. Each one is a daughter chromosome. They have only one chromatid each. But here the parent chromosome, one chromosome, it has two chromatids. So in the anaphase beginning, each chromosome will be having two chromatids and they are called sister chromatids anyhow. And in the anaphase, the centromere will be splitting exactly in the middle so that it results in the formation of daughter chromosomes, and daughter chromosomes are having only one chromatid each. This is the first event which will happen in the anaphase. After that, the second event will be the movement of these particular daughter chromosomes. They migrate. Migration of daughter chromosomes to the respective poles, to opposite poles. So when they are moving to the opposite poles, they will be taking the shapes of V, L, J and I shapes. So the shapes of the chromosome, if they ask the shape of the chromosome, shapes of the chromosomes can be seen. Shapes of chromosomes can be seen. In which phase? In anaphase. And they'll be taking the shapes of V, L, J, I shapes. So two main events of the anaphase will be centromere splits results in the formation of daughter chromosomes, and daughter chromosomes will be having only one chromatid each. Then the daughter chromosomes which are formed, they will be pulled towards the respective poles with the help of the spindle fibers, and they'll be migrating towards the opposite poles. When they're moving, they will be taking the shapes of V, L, J, I, so that we can see the shapes of the chromosomes in anaphase. Next is the telophase. Telophase. This is exactly opposite to that of the prophase. All the events will be opposite to prophase. Let us see the first event, what will happen in the prophase. We have started with the condensation of chromatin will begin. Here, decondensation of chromosomes. Decondensation of chromosomes into chromatin. Decondensation of chromosomes into chromatin. That means before the chromosomes were this much size, now they are uncoiled and they are hydrated and they'll become elongated structures like chromatin. That will happen here first. Later, around them, the nucleolus and nuclear envelope, ER and Golgi reform. So this event is also exactly opposite to that of the prophase. By the end of this, we'll be getting 2 daughter nuclei. Results in the formation of 2 daughter nuclei. But only 2 daughter nuclei are formed. So this telophase will be continued in terms of cytokinesis. The cytokinesis in plants occurs by cell plate method. Whereas in animal cells, it occurs by furrow formation. Furrow formation. This in case of plant cells, this in case of animal cells. What you have to remember about cytokinesis is cytoplasm will be dividing around the 2 daughter nuclei and it will form the 2 daughter cells. And in the cytokinesis of the plant cell, cell plate will be the first structure. Precursor of the cell wall will be the cell plate. It is formed from the Golgi vesicles and vacuoles. And the cell plate will be growing, from the center towards the side. So it will be growing centrifugally in plant cells. Cell plate will be growing centrifugally. That point you have to remember. Whereas in animal cells, furrow will be formed on the sides and it will proceed towards the center. So furrow will be growing towards the center. So centripetal manner it will be growing and it divides one parent cell into 2 daughter cells. So ultimately, after the mitosis, the complete mitosis, which happens by karyokinesis and cytokinesis, we'll be getting 2 daughter cells which are identical in all aspects to that of the parent cell and among themselves also. They are similar among themselves with other daughter cells also and they are similar to the parent cells also. As the daughter cells are similar to that of the parent cell in all the aspects like chromosome number, chromosome structure, genes content, everything, this type of mitotic division is also called as equational division. And the mitosis significance is, it is important for the growth of the cell. Growth in the sense, one organism is growing because of the cell multiplication. So in multicellular organisms, it is involved in the growth of the cells. In unicellular organisms, it is involved in the growth and reproduction of the cells. And in multicellular organisms, it is concerned with the cell repair. The damaged cells will be replaced by the mitosis. So replacement is also a function. Nucleocytoplasmic ratio is also maintained by the mitosis. So this is the significance of mitosis. Let us see the MCQs now. Number of mitotic divisions required to produce 128 cells from a cell is. From one cell is given to you. And this should be forming 128 cells. There is a formula for this if you want to calculate the number of mitotic generations. 2 power n is a formula. But here, how many cells are undergoing division? That is the question here. So, how many mitotic divisions are required to form 128 cells? So, one cell first it will be forming 2 cells here. 2 will be forming 4. Like this, every time it will be doubled. 8, 16, 32, 64 and 128. But this one will be giving you 2 power n will be giving you number of mitotic generations. So if the question is asked for mitotic generations, obviously the number will be 1, 2, 3, 4, 5, 6 and 7. 7 generations it will be taking to form the 128 cells. But in the given options, there is no 7. So, they are talking about the mitotic divisions occurring in the cells. So, how many cells are actually undergoing the mitotic divisions to form the 128 cells means there is a formula for this question. For that, formula is n minus 1. N is number of daughter cells. So how many daughter cells are there? The formed cells are 128. So 128 minus 1, 127. So number of mitotic divisions required to form 128 cells will be 127, because you have to use the formula n minus 1. So for this question, the correct answer will be 127. Next one. The products of mitosis are. What are the products that we have got by the end of the mitosis? Two daughter cells are there, which are identical in all aspects. So two genetically identical cells, one nucleus containing twice as much as DNA as parent, one nucleus having twice as much as DNA as parent is not there in this. Two genetically identical nuclei. Nuclei will be formed only at the end of the telophase. But the question is asked about the mitosis end products. Four nuclei containing half. Four nuclei are never formed in the mitosis, because four nuclei will be there in the meiosis, not in the mitosis. So for this question, the correct answer will be A. Two genetically identical cells which are similar among themselves and similar to that of the parent cell in terms of number of chromosomes, in terms of DNA content, everything. Because whatever the DNA that got doubled in the S phase, okay, if you take the DNA content is 2C before S phase, in the S phase it will become the 4C. And then in the two daughter cells, each daughter cell will be getting the 2C, 2C. So the two genetically identical cells will be formed at the end of the mitosis. Mitotic anaphase differs from metaphase in possessing. You have to identify first among the given characters, anaphase characters. Same number of chromosomes and half number of chromatids. Same number of chromosomes and half number of chromatids. Anaphase characters will be double the number of chromosomes but same number of chromatids. Because in anaphase, this type of chromosome splits up into daughter chromosomes. Chromatids remain same. Here also two chromatids, here also two chromatids only. But number of chromosomes, here it is only one chromosome. Here it is two daughter chromosomes. So by knowing that concept, this is wrong. Same number of chromosomes, same number of chromatids is wrong. Same number of chromosomes and half number of chromatids is also wrong. Half number of chromosomes and half number of chromatids is also wrong. Double number of chromosomes and same number of chromatids is correct. For this question, double number of chromosomes will be there because daughter chromosomes are formed, and same number of chromatids. Here also two chromatids, there also two chromatids. So for this question, the correct option would be double number of chromosomes and same number of chromatids. At the end of this phase, the cell does not show ER, Golgi, nucleolus and nuclear envelope. At the end of this phase. At the end of which phase all of them disappear. Anaphase, telophase, prophase, metaphase. You know that it is prophase because they are formed in the telophase, reformed in the telophase, they disappear in the prophase. Metaphase is marked by initiation of condensation of chromosomal material. This happens in the prophase. Chromosomes align at the equator. This happens in the metaphase. Disappearance of ER and Golgi. This also occurs in the prophase only. At the end of the prophase. Initiating the disintegration of the nuclear membrane. This also happens at the end of the prophase. End of the prophase. So here, for this question, the correct answer will be chromosomes align at the equator. This is the correct option because they are all arranged in the center or equator to form the equatorial plate or metaphase plate. What is the significance of mitosis? The significance of mitosis, growth is there, repair is there, and replacement of the cells is also there. Growth will happen in the multicellular organisms and unicellular organisms because of mitosis. Cellular repair, damaged cells will be repaired by the mitosis only. And in the place of old cells, old worn out cells, the new cells will be replacing the old ones by the mitosis. So all these three are the functions of mitosis. So for this question, correct answer will be all of the above. Major event that occurs during anaphase which brings about equal distribution of chromosomes is during anaphase which brings about equal distribution of chromosomes. What is a major event? You know, in anaphase, centromere splits and the daughter chromosomes will migrate towards the opposite poles. So let us see in these options which is correct. Condensation of chromatin. Condensation of chromatin occurs in the prophase. Splitting of chromatids. This occurs, chromatids will not split. Basically, splitting of centromere will be happening. DNA replication. This occurs in the S phase of the interphase. Then splitting of centromeres occurs in the anaphase only. This is important for the equal distribution of chromosomes. So for this, the correct option will be splitting of centromeres. This is the major event which will happen in the anaphase, that is meant for the equal distribution of genetic material. Next one. If there are 10 power 5 cells per ml growing in every 35 minutes, how many cells will be there after 175 minutes? So basically 35 minutes, the cell will be multiplying and it is doubling for every 35 minutes. So in 175 minutes, how many cells will be there in the medium? So out of 175 minutes, okay, 35 minutes means how many times it will be dividing? 5 times it will be dividing 175. So 5 times is the answer. The cells will be divided. But initially, how many cells are there? 10 power 5 cells are there. So there is a formula for this to calculate the number of cells, that is 2 power n into number of given cells, where n is nothing but n is number of times. Number of times the divisions. So here the 2 power n will be 2 power 5 into 10 power 5. 2 power 5 will be 32. So 32 into 10 power 5 cells. So for this question, the correct answer will be 32 into 10 power 5 cells. So the time which is required, okay, to form 32 into 10 power 5 cells will be 175 minutes. So the answer for this question, as we have got that, it is 32 into 10 power 5 cells. This is how you are supposed to do. You have to use the formula 2 power n into given cells. That given cells will be given already there. 10 power 5 cells are given. 2 power n, where n represents the number of times the cells are dividing. That you need to calculate like this. Next one. A mutation in a bacterial cell results in non-formation of mesosomes. The expected results would be non-formation of mesosomes. Mesosomes are not formed because of the mutation. And the expected results would be, what will happen in that case? Cell division will not occur. Definitely it will not occur. Replication of DNA also will not occur. Karyokinesis also will not occur. Basically, karyokinesis means nuclear division. Here there is no nuclear division. This will also not happening. So all of them will not be there. So all the above will be correct for this. A mutation in a bacterial cell, if it is not forming the mesosome, cell division cannot complete, replication cannot complete, and nuclear formation also cannot be completed. Suckers are produced by. Suckers are asexual reproductive structures. Or vegetative propagules. So that suckers will be multiplying by, they are produced by parthenocarpy. This is nothing but the formation of fruit without fertilization. Parthenogenesis, formation of the organism without fertilization, just from the gamete. Female gamete will be developing into the organism. Mitosis and meiosis. You know that cell growth, multicellular organisms or unicellular organisms will be growing by the mitosis only. So here vegetative propagules or asexual reproduction structures are all formed by the mitosis only, never by meiosis. So for this question, rhizome, corm or stolon, suckers, all these things are produced by the mitotic divisions. Which of the following phase is not a part of the interphase? You know, interphase is having G1, S and G2 phases. And which is not a part of this. S phase, anyhow it is a part, G1 is a phase and G2 is also a phase. M phase is not a phase of the interphase, because M phase is the mitotic phase or divisional phase. This is nothing but mitosis phase. This is never a part of interphase, but both of them together, interphase and M phase, will become the part of cell cycle. Match column 1 with column 2 and select the correct option. Column 1, disintegration of the nuclear envelope. Disintegration of the nuclear envelope occurs in which phase? The disintegration will happen in the prophase. Appearance of nucleolus will happen in the telophase. Division of centromere, it occurs in the anaphase. Replication of DNA, it occurs in the S phase. So for this, nuclear envelope will be disappearing. Nuclear envelope, nucleolus, Golgi, ER, they disappear in prophase and they reappear in telophase. Centromere will be dividing into two daughter chromosomes in the anaphase. Then replication of DNA occurs in the S phase. So for this question, the correct answer will be the C, A2, B3, C1 and D4. Next one. One cell divides every minute. In 20 minutes, these cells filled the culture tube. One complete tube got filled by that. How much time is required to fill half of the tube? How much time is required to fill the half of the tube is, like in 20 minutes, one full tube. And for every minute it is doubling. So the doubling time is just 1 minute. So if it is full tube in 20 minutes, before that minute, okay, it would have filled half of the cup or half of the tube. So minus 1, it will be 19 minutes. If you do minus 1 one more time, it would have become half of this half, that is 1/4 tube. So for every minute, if you take that 18 minutes, 1/4 tube is there, then in 1 minute, it would have filled half of the tube. In the next minutes itself, that is 20 minutes itself, this half would have become the full tube. So this is how you need to calculate. Just you have to do the minus 1 because the question is asked to fill half of the tube. So half of the tube means you will be getting the answer for this is 19 minutes. One tube in 20 minutes, so half tube in 19 minutes, n minus 1. It is important that the centromere do not divide till the end of the metaphase because why it is not dividing at the metaphase, why it is dividing only in the anaphase? Because it is connected with the nuclear membrane, it produces spindle fibers, contains genes that control prophase, holds the replicated DNA molecules together. Because in the metaphase, the centromere is holding the replicated DNA molecules together, or you can think that chromatids are holding together because of the centromere. The replicated DNA molecules are nothing but the chromatids only. So it is holding up the chromatids, two sister chromatids together, and they are not dividing. So for this question, the correct answer will be holds the replicated DNA molecules together. That's all. So all of you prepare well for the exams. Thank you.